Yeah, it doesn't like green in the center. It does like green in the surround, but it doesn't like red in the surround, right? So it's got some of what it likes in the surround, but it's also got a fair bit of what it doesn't like, right? So overall, this is the worst position of this in this general area for this cell. Obviously, the worst overall would be a green spot surrounded by red, but this is the closest we can come to that for what's on the screen right here. And the best position, again, on this screen here would be this, right? Because this comes closest to what the cell prefers, which is all red in the center, or long wavelengths in the center, no green wavelengths in the center, and it doesn't like red in the surround, and it does like green in the surround, right? So it's got some green that it likes in the surround. It's also got some of what it doesn't like, but overall it still likes this the best of anywhere this cell's receptive field could be on the screen, right? So again, the totally best stimulus for this cell would be this, but this is kind of as close as we can come to that on this particular screen. Okay, I know this is a little kind of weird and complicated to think through, like how this works, but do you guys have questions? Does that make sense to you? You hanging with us okay so far, how these receptive fields would work? Okay, so that's single opponent versus double opponent kind of receptive fields. So, you know, essentially, we end up with kind of a sort of, you know, the double opponent gives us kind of the receptive field that we saw with these LGN cells and with the retinal ganglion cells for just brightness. It just takes more steps essentially, right? So because essentially the idea is here, before we can get a cell that prefers this, we kind of have to have an earlier stage of processing where this happens. And you know, the good news for you guys is we're not gonna really like fuss too much about figuring out how that's wired up, but just trust me on that, that kind of like, we can't go directly to this as easily as we can when we're talking about pure brightness. You kind of have to have this single opponent sort of stage of processing before you get double opponent cells. Now these single opponent cells, so when we're talking about where they are, you would mostly find single opponent cells like this in the retina and the LGN. So, you know, again, when we're talking about just brightness or achromatic stuff, we saw receptive fields that kind of look like this for achromatic stuff in the retina and LGN. But in the retina and LGN for color, we really only see kind of these single opponent cells. It's not actually until a later stage of processing in like the primary visual cortex that we get these double opponent cells, right? So color kind of sort of takes a longer pathway in the nervous system to kind of get to receptive fields that look like this, right? So color is kind of like one synapse behind just pure brightness in terms of like where this kind of receptive field emerges in the nervous system. Now, if you guys remember, we talked a little bit when we talked about like hypercolumns and the ice cube model and all that stuff. We talked about these kind of chunks of cells kind of interspersed among all the other stuff in the hypercolumns that we called blobs that were filled with color-sensitive cells. And at least sort of the conventional wisdom is that those blobs would have cells like this, these double opponent cells in them. Now, as usual, like when you dip into the science a little deeper, there's lots of arguing about how much of that is exactly true and how much that's oversimplified and so on. But for the purposes of this class, we can at least pretend or imagine with some hand waving that it's as simple as that, and that like these are the kinds of cells that you would find in the blobs of the primary visual cortex, and these are what you would find in like retina and the LGN. Okay. Questions about that so far? All right, so the key concepts here are that you have these single and double opponent cells in your nervous system, right? Now, so what this gives us, right, is cells that have like a red-green contrast, right? So I kind of foreshadowed this before. Essentially what that means, right, is you have, this is a little bit more of an abstract or kind of an overarching view of how this works in the nervous system. So essentially the idea here, right, is that what this gives us in the case of the receptive fields we just saw on the last slide is certain cells in the nervous system that have receptive fields like that, you know, like red in the center and green in the surround. You also have obviously the opposite of that, right? So you will have certain cells in the early stages of visual processing that, you know, essentially are sensitive to red versus green contrast, right? So those receptive fields we looked at on the previous slide, we're only getting input from like the red and the green cone sort of neural pathways, right? They didn't get any input from the blue cone pathway at all, right? So we had cells there that were like only concerned with the red versus greenness of their receptive fields. They were kind of like agnostic, or they didn't care about how much blue light was kind of falling, right? So essentially, that gives us like one of the color opponent pathways we have in our nervous system is like based on red versus green contrast.